everyone. This is Sungmin Kang from Stanford University. It's an honor to have an opportunity to speak today. My talk will be on the latest trend and recent research in the field of aging. As aging is such a broad field of study, recent trend can be divided into 12 aging hallmarks with three main categories: primary, antagonistic, and integrative. First, the primary hallmark reflects damages affecting genome, telomere, and epigenome, which directly affects the aging process. And the antagonistic hallmarks are best described as a double-sided sword. For example, sufficient nutrient intake or the activation of mitochondria is advantageous in use. However, as we age, the same traits can lead to the excessive ROS accumulation or inflammation, contributing to the aging process. Lastly, the integrative hallmarks arise when the accumulated damage caused by the primary and the antagonistic hallmarks cannot be compensated anymore, resulting in the stem cell exhaustion or chronic inflammation, which dictate the pace of aging. So based on these aging hallmarks, my research has been focused on two of them, which are the genomic instability and epigenetic alterations affecting muscle stem cell aging. The muscle stem cells, also known as satellite cells, reside between the fibers inside the basal lamina. And these cells remain quiescent under normal conditions. However, when muscle damage or injury occurs, these cells are activated and re-enter the cell cycle to generate muscle progenitor cells, facilitating the regeneration of new muscle fibers. Additionally, they have the ability to self-renew, ensuring the replenishment of the stem cell population. So it's been reported that during aging, satellite cells lose their reversible quiescent state and transition into a senescence-like state, which negatively affects the regeneration process, impairing activation, proliferation, and self-renewal capabilities. So consequently, as individual age, the functionality and the quantity of satellite cell decline, contributing to skeletal muscle aging. Then how can we effectively reverse the aging process in adult stem cells? The epigenetic lens model has been proposed to explain why the epigenome is one of the key targets for rejuvenating interventions. In this model, this genome represents genomic information, and the lens represents epigenome, and the projected image represents the phenotype. With age, there is a deterioration of cellular phenotype, and this may be due to the intrinsic changes of DNA as well as the epigenome. Although correcting the altered DNA information could be difficult, we can easily change the epigenetic modifications. So if we restore this epigenetic lens toward more youthful state, the aged phenotype also could be partially restored. So among several different epigenetic changes during aging, the global loss of heterochromatin has been observed in all eukaryotes, and it's known as a cause of aging. So aged cell shows the reduced level of histone HPK9 trimethylation and ATP1 compared to those young cells, resulting in more relaxed chromatin structure, which is predictive of genomic instability, including higher levels of DNA damages and the expression of aberrant transcripts. However, the cause of this loss of heterochromatin has remained enigmatic. In order to directly assess whether heterochromatin loss occurs during muscle stem cell aging, we use the electron microscopy to measure the heterochromatin. And indeed, we found a significant decrease in the heterochromatin level in muscle stem cells from old mice compared to young mice. So based on this, we pursued the investigation to uncover the cause of heterochromatin loss during aging. It's been well documented that the metabolism directly affects chromatin structure by altering epigenetic modifications. Here are some examples that how several metabolites are used in epigenetic modifications as essential cofactors. For example, the S-adenosine methionine, which is a metabolic intermediate of one carbon cycle, is a methyl donor for histone and DNA methylation. And the acetyl-CoA, which is generated from TCA cycle, is an acyl donor for histone acetylation. So these imply a direct interrelation between metabolism and epigenetic modifications. Since the heterochromatin formation is associated with the histone H3K9 methylation, we decided to measure the S-adenosine methionine in young and old muscle stem cells. So from this untargeted metabolomics, we found that DCSAM and MTA increase, whereas SAM and SAW decrease with age. When SAM is used in methylation reaction, it generates methylated product and yields SAW. Whereas when SAM is converted to decarboxylated SAM, it can be used in polyamine synthesis and yields MTA. Although SAM is widely known as just a methyl donor, actually it is also the only amine donor for synthesizing polyamines, which indicates that excessive consumption of SAM in polyamine synthesis may reduce its availability for transmethylation. So based on this premise, we raised two questions. First, would depleting SAM induce loss of heterochromatin by reducing H3K9 methylation? And second, does an increase in spermidine synthesis drive SAM depletion in aged muscle stem cells? Consistent with this premise, we discovered that polyamine themselves, including the spermidine and putrescine, increase with age, whereas the precursors of polyamine synthesis, including ornithine and arginine, decline with age in muscle stem cells. To understand why spermidine synthesis increases in aged muscle stem cells, we quantified the level of enzymes related to polyamine metabolism. And this revealed that the expression of SRM and PAOX increased, whereas that of the SET1 decreased with age. As SRM, the spermidine synthase is an enzyme that directly consumes the DCSM. We hypothesized that the high level of SRM expression leads to increased use of SAM in polyamine synthesis and results in SAM depletion in aged muscle stem cells. To address this, we first confirmed intracellular SAM levels in muscle stem cells. So consistent with previous data, the intracellular SAM decreased with age, and the intraperitoneal injection of SAM successfully restored the SAM levels in old muscle stem cells. So we tested whether inhibition of the SRM could restore SAM contents in aged muscle stem cells. To do that, aged mice were treated with MCHA, which is a specific inhibitor of spermidine synthase, and the intracellular SAM and spermidine levels were quantified. So we found that inhibiting SRM not only decreased the spermidine level, but also significantly increased the SAM content. So this suggests that inhibiting spermidine synthesis is able to restore SAM content in old muscle stem cells. To test whether increased SAM availability induces heterochromatin formation, the levels of H3K9 trimethylation and ATP1, which are two markers of heterochromatin, were assessed in muscle stem cells in muscle tissue sections from young mice, old mice, and old mice treated with SAM or MCHA. And we found that heterochromatin markers decrease with age, and the SAM or MCHA significantly restores the level of markers in old muscle stem cells. As the loss of beta-carotene is associated with enhanced chromatin accessibility, we conducted a TEC-6 on muscle stem cells isolated from its group of mice. So comparisons of the ATEC peaks across the genome showed more than 3,000 peaks showing increased accessibility in aged muscle stem cells, which were partially rescued by the SAM or MCHA treatment. And these representative summit-centered heat maps show higher signals in aged muscle stem cells compared to those in young muscle stem cells, which were rescued by SAM or MCHA. So these results are consistent with the premise that loss of heterochromatin is accompanied by an increase in chromatin accessibility during aging of muscle stem cells. And it's been well known that increased expression of transposable elements because of the heterochromatin loss leads to the DNA damage and during aging. So we measure the mRNA expression of TEs, including line, sign B1, and sign B2. And there was a significant increase in the expression of TEs in aged muscle stem cells, and this was rescued by SAM or MCHA. The previous works from our lab and others show that the loss of heterochromatin renders the genome more susceptible to DNA damage. In order to test whether aged muscle stem cells are more susceptible to genotoxic stress, we isolated cells from its group of mice that had been subjected to X-ray irradiation and quantified the extent of DNA damage. By quantifying the number of gamma H2AX foci or the cells with cometel, we found that with age there was an increase in DNA damage, and the SAM or MCHA partially reduced the DNA damage in old muscle stem cells. In addition, we tested whether the increased susceptibility to genotoxic stress leads to the cell death. So the dead cells were measured by tunnel MPI staining from muscle stem cells isolated from its group of mice, followed by 2 days of ex vivo activation. So these analyses revealed a twofold increase in the number of activated muscle stem cells that are dying in old mice compared to young mice, which were partially rescued by restoration of SAM. Since the MCHA is able to increase SAM at the same time decrease pyrimidine, we wanted to test whether the beneficial effects we observed are due to the decrease of pyrimidine or the restoration of SAM by testing the effects of it independently. First, to test whether it's due to the decrease in SAM, we prevented the decrease in spermidine, we prevented the increase in SAM in MCHA-treated cells by treating them with cycleucine, which is an inhibitor of methionine adenosyltransferase. So, treatment of MCHA-treated cells with cycleucine leads to a decrease in spermidine without an increase in SAM levels. And conversely, to test whether the beneficial effects were specifically due to an increase in SAM, we prevented the decrease in spermidine in MCHA-treated cells by adding the exogenous spermidine. So treatment of the cells with spermidine concomitantly with MCHA leads to an increase in SAM without a decrease in spermidine levels. So with this ability to independently assess the effects of increasing SAM and decreasing spermidine, we quantified the extent of the DNA damage and cell death. So cells treated with both MCHA and cycleucine exhibited greater DNA damage and increased cell death compared to the MCHA alone, suggesting that the beneficial effects of SRM inhibition are due to the increase in SAM levels. By contrast, the preservation of spermidine in MCHA-treated cells showed that the benefits are similar to what we observed in MCHA alone. indicating that a decrease in spermidine does not account for the beneficial effects of SRM inhibition in aged muscle stem cells. Additionally, as AMD1 promotes SAM consumption during the polyamine synthesis, we tested whether inhibiting AMD1 redirects SAM to the transmethylation. As expected, treating aged muscle stem cells with MGBG, which is a specific inhibitor of AMD1, resulted in a decrease in spermidine and an increase in SAM. And we observed significantly increased heterochromatin markers and reduced the DNA damage accumulation and the cell death in MGBG-treated aged muscle stem cells. So these data support our conclusion that restoration of SAM leads to protection against the DNA damage in aged muscle stem cells. Then to further investigate whether the protective effects of SAM restoration is due to the reformation of heterochromatin, the H3K9 methylation was inhibited by treating cells with inhibitors of methyltransferases. The UNC0642 is a specific inhibitor of G9A and GLP, which are the enzymes for catalyzing mono and dimethylation of H3K9. And the cetirizine is an inhibitor of sub39H1 and G9A, which has been known for its ability of unpacking heterochromatin. So we first knocked down the spermidine synthase using the siRNA and treated cells with its inhibitor. The knocking down of SRM restores the heterochromatin markers, which were significantly ablated by the methyltransferase inhibitors. Then to promote the DNA damage, we treated its group of cells with doxorubicin before the quantifying the DNA damage and cell death. And as expected, we observed that knocking down of SRM leads to dramatic reduction in the number of DNA damaged foci and the cell death, and this effect was partially abolished by inhibiting H3K9 methylation. We also confirmed the effect of SAM restoration in isolated human muscle stem cells. So directly treating human muscle stem cells with SAM or MCHA increased the heterochromatin markers, which were decreased following methyltransferase inhibitor treatment. And the DNA damage and cell death were also quantified from human muscle stem cells with genotoxic stress in culture. So as expected, we found that MCHA reduced the DNA damage and cell death, and these effects were prevented by the treatment with methyltransferase inhibitors. Then we tested the cell intrinsic effects of SAM restoration on muscle stem cell potency to regenerate new muscle fibers by conducting muscle stem cell transplantations. So its group of cells were transduced with the lentivirus expressing luciferase, and then the cells were transplanted into the immunocompromised mice with the barium chloride injury. So the data shows that the muscles transplanted with SAM or MCHA-treated cells showed higher signals than those with vehicle-treated cells, indicating that the restoration of SAM improved the tissue regenerative potential of aged muscle stem cells. Then to track the fate of transplanted cells, we used the muscle stem cells isolated from aged PEG7-CRE RFP mice, where administration of tamoxifen induces the expression of RFP as a reporter specifically in muscle stem cells. So RFP positive muscle stem cells were transplanted and the muscles were collected 2 days after transplantation, and the RFP labeled muscle stem cells were counted using FACS. We found a significantly higher number of RFP positive muscle stem cells in muscles transplanted with SAM or MCHA treated cells compared to those with vehicle treated cells. So given the 2 days of short time of muscle stem cell re-isolation after transplantation, the number of the RFP positive muscle stem cells is most probably the result of survival rather than proliferation. So this data demonstrate that the effect of cell restoration on cell survival is associated with improved capacity of aged muscle stem cells to regenerate new muscle fibers. And to further test whether the improved muscle regenerative potential was due to the increased heterochromatin levels, we performed 2 additional transplantations. First, the aged muscle stem cells were transduced with either a scrambled shRNA or SRM shRNA. And consistent with the data from MCHA-treated cells, the SRM deficient cells contributed more to the formation of new muscle fibers. And to assess whether formation of ATPase trimethylation leads to enhanced regenerative potency of SRM deficient muscle stem cells, the cells were transduced with shRNA targeting SRM alone or both SRM and Sub39H1. And interestingly, we found that muscles transplanted with both SRM and Sub39H1 deficient muscle stem cells showed much lower signals compared to those with SRM-only deficient muscle stem cells. Collectively, these data support our previous finding that increasing ATPase trimethylation by inhibiting spermidine synthesis rescues the impaired muscle regenerative capacity of aged muscle stem cells. We next wanted to determine whether the reduced DNA damage and cell death are associated with improved muscle regeneration of aged mice. To address this question, we first tested whether SAM or MCHA treatment reduces the DNA damage and cell death during in vivo activation at 2 days after muscle injury. So we found that aged mice show a higher level of DNA damage and cell death compared to young mice, which was rescued by SAM or MCHA. And finally, to determine whether the reduced DNA damage and cell death are associated with the improved muscle regeneration, we collected muscles from each group of mice 7 days post-injury and examined the regeneration histologically. So by measuring the regenerating myofiber cross-sectional area, we found that muscle regeneration decreased with age, but the treatment of aged mice with SAM or MCHA enhanced the regeneration efficacy similar to the level of young mice. And to confirm that the improved muscle regeneration is associated with increased muscle stem cell survival, we measured the total PAX7 positive muscle stem cells after muscle injury. And we found that a significant decrease in the number of muscle stem cells in aged muscle compared to young muscle. And this was partially rescued by SAM or MCHA. These data imply that impaired muscle regeneration during aging is associated with increased DNA damage and cell death, and this can be partially rescued by restoration of SAM. Lastly, to assess functional recovery from SAM restoration, we measure the muscle force using a grip test after muscle injury. We found that aged mice exhibited lower grip strength compared to the young mice, and this was partially rescued by restoration of SAM. In addition, we monitored the gait, which is the pattern of walking, 14 days after muscle injury, and observed that aged mice showed significantly delayed average duration of one gait cycle compared to that of young mice, which was also partially rescued by Sam or MCHA. So these data suggest that heterochromatin reformation could eventually improve the functional recovery of aged mice. So to summarize, this work aims to answer the fundamental question of why heterochromatin declines with age. So we showed that reduced intracellular SAM availability decreases the H3K9 methylation and heterochromatin formation in aged muscle stem cells. We further demonstrated that the systemic restoration of SAM in old muscle stem cells to a useful state induces the heterochromatin reformation and rescues many age-associated phenotypes, including DNA damage, susceptibility to genotoxic stress, and delayed muscle regeneration. So I'd like to express my gratitude to all the members of Randall Lab and Stanford and my mentor Tom for their unwavering support. Thank you for your attention, and please feel free to ask any questions. I'm more than happy to address them. Thank you.